Hi, my name is Li Taifang. I'm a scientist at Endpoint Health. Welcome to this virtual presentation. I'm presenting on behalf of the somatic mutation working group of the SEQC2 consortium for our work to establish a pair of tumor normal somatic mutation reference samples. This work took place when I was at Roche Sequencing Solutions. This slide shows the karyotyping of these two cell lines, the tumor on the left and the normal on the right. On the normal karyotyping on the right, you can see the 46 chromosomes in a human cell. Most of these chromosomes look quite normal, except for the rearrangement that fuses part of the chromosome 6 with chromosome 16 in this blow-up figure toward the bottom. For the tumor karyotyping on the left, however, it shows 66 chromosomes. Many of those chromosomes are highly rearranged and unrecognizable. This results in a diverse set of copy number states across the genome that will result in somatic mutations of different variant allele frequencies, or VAF. This slide shows the histograms of the variant allele frequencies for all the somatic mutations in our high confidence reference call set. The SNVs are on the top and the indels are on the bottom. It is quite obvious that there are multiple peaks for the SNVs VAFs at around 0.67, 0.5, 0.33. These fractions correspond to VAFs from different parts of the genome. In this figure, the different colored regions represent different copy number states. VAFs shift alongside the color shift. When the color shifts, the VAF shifts along with it. This is a way to show that the VAF shifts are consistent with color changes, or the VAF are consistent with their copy number states. Low VAF variants indicate subclonal mutations. This slide shows another way to represent the same thing. We group all the somatic mutations in regions of different copy numbers into each single histogram. Their peaks are consistent with their copy number states. For example, this subfigure on the right shows that the VAF histograms for somatic mutations in regions where the copy number is 2. You can see peaks at 2/2 and another peak at 1/2. This subfigure on the middle left is from copy numbers of 3, and you can see clear peaks at 3/3, 2/3, and 1/3. The VAFs are consistent with the copy number states. In addition, however, there are always low VAF variants, and they correspond to subclonal mutations. This is the bioinformatic workflow we have used to establish the high-confidence somatic mutation reference call set. First, we started with 21 whole genome sequencing replicates from 6 different sequencing centers. Each of these 21 sequencing replicates were aligned with 3 different aligners to create 63 tumor normal band files. For each of the 63 tumor normal band files, 6 mutation colors were run. Then, we used 2 machine learning algorithms, somatic-seq and usomatic, to classify mutations in each of these 63 datasets based on color classifications and sequencing features. Initial confidence levels were assigned based on each mutation call's reproducibility across different aligners, different sequencing centers, and different sequencing replicates. In short, the high confidence mutations have support in majority of the sequencing replicates, in majority of the sequencing centers, and majority of the aligners. However, low VAF somatic mutations will not be highly reproducible due to sampling. For those, we use 3 additional datasets to rescue them back into the reference call set. By combining the 9 NovaSeq replicates from Illumina into a single 400x dataset, and an additional independent 350x dataset from Genentech. Furthermore, we combine all the whole genome sequencing data we had into a single 1500x dataset. Each of those high-depth data were also classified by somatic-seq and new-somatic across 3 different aligners in order to make high-confident calls out of low VAF variants. Finally, we used PacBio long-read data to remove a small number of mutation calls in low mapability regions when short-read and long-read data contradicted. This is an illustration of how we trained the classifier. We took advantage of multiple sequencing replicates of the normal cell line to build machine learning classifiers. A normal replicate would be designated as the normal, and another normal replicate would be designated as the tumor. We spiked in silico mutations into the designated tumor by randomly changing the base call of some reads. We aligned those altered reads. This workflow was done by BAM surgeon. Then we ran the somatic mutation workflow. We used the spiked in as the ground truth. Everything else that was called as a somatic mutation was a false positive. For somatic-seq, we built a classifier for each sequencing platform and each sequencing center. For new somatic, all training data were combined, so we had 3 new somatic classifier, 1 for each aligner. This shows the reproducibility characteristics of the 4 confidence labels. For high conf calls, on average, there are more than 60 pass calls and fewer than 1 reject calls. Those tend to be high VAF variants that are almost universally reproduced. For metconf calls, there tend to be almost half of the examples where they are called as pass, but fewer than 1 reject. They are mostly low VAF variants that are not highly reproduced across many replicates, but they also don't have the kind of sequencing features that will make them false positives. For low conf calls, however, the reproducibility is too low to confidently distinguish them from noise. And then for the unclassified calls, there are more rejects than pass calls. They are likely false positives due to mapping errors, germline signals, or from uncallable regions. Positions around low conf and unclassified calls were removed from the high confidence region. High conf and metconf calls are grouped as the reference call set. This shows you the breakdown of the 4 confidence labels. Variants labeled as high conf and metconf are regarded as true positives and are included in the high confidence call set. This is the 3D scatter plot for somatic-seq classifications. For each somatic variant call, the x-axis is the number of times somatic-seq has classified as a pass or a likely true mutation out of 63 datasets. The y-axis is the number of times somatic-seq has classified it as a reject or a likely false positive out of the 63 datasets. The z-axis is its variant allele frequency, again VAF. Just a note, the number of pass and reject won't always add up to 63, because if a variant is not called by any of the 6 callers, then somatic-seq won't have a chance to classify it either way. If there are many variants in the middle of the XY plane, it means that there are many calls where somatic-seq disagree with its status from replicate to replicate. In SMV, however, you hardly see them. That means somatic-seq is very consistent across different sequencing replicates. For high-conf and mid-conf calls, you hardly see any reject classifications, even though here in the shades of blue, blue is the color for mid-conf calls, the number of passes are rather low. They are such because when somatic-seq has a chance to make a decision on them, it's always a pass. But since they are low VAF, they do not always appear in each replicate. The black bar dots are unclassified calls. Some of them are repeatedly detected by the callers, but repeatedly classified as reject across the VAF spectrum, mostly due to low mapping quality, misalignment, or other sequencing features that indicate that they are false positives. In indels, however, there are some calls in the middle of the XY plane, indicating inconsistent somatic-seq classifications. We did some manual inspections though. We learned that almost all of them are long deletions but true indels. They are not always called as a pass because they carry some features such as soft clip reads that confuse somatic-seq into classifying them as false positives sometimes. Their labels were manually changed to high conf. These two figures show the very high agreement between somatic-seq and new-somatic classifiers. Keep in mind that somatic-seq and new-somatic use totally different machine learning algorithms. Somatic-seq uses adaptive boosting trees, whereas new-somatic uses deep convolutionary neural network. The figure on the left is a scatter plot for each mutation call. The x-axis is the number of times out of a total of 63 that a mutation has been labeled as a pass by somatic-seq. The y-axis is the number of times the same mutation has been labeled as a pass by new-somatic. The z-axis is its VAF. Almost all of the data points on XY plane falls right onto the perfect XY diagonal line, visually demonstrating that somatic-seq and new-somatic almost always agree. The z-axis shows that whereas mid-conf and low-conf calls are usually low VAF, both high-conf and unclassified calls have a full spectrum of VAFs, again demonstrating that for unclassified calls, they were labeled as likely false positive not due to them being low VAF, but for having sequencing features that indicate that they're likely false positives. For the two figures on the right, they show histograms for all the somatic mutations, how many times they are classified as a likely mutation by the two machine learning classifiers. For the vast majority of them, in both somatic-seq and new-somatic, the numbers are either above 60 or below 10. In other words, for the overwhelming majority of variant calls, they are either highly reproduced as pass calls or almost none at all. There is very little conflict between the replicates. These four figures show the scatter plots of the four orthogonal validation platforms. The top left figure shows the VAF scatter plot for a set of randomly chosen variant calls. The x-axis shows the VAF estimated from the reference whole genome sequencing datasets. The y-axis shows the VAF estimated from the 2500X AmpliSeq data sequenced on the MySeq. Solid circle means that the calls are validated following a set of validation rules. The validation rules state that there is clear variant signal in the tumor and no variant signal in the normal. The open circles are for variant calls that were not validated. which means there was either no variant read despite high coverage, or if there was germline signal in the matched normal. Almost all of the green data points, those are the high conf calls, are within the 95% confidence interval indicated by the diagonal oval shape in the middle, indicate that their VAF estimates are the same for both platforms. The Pearson's R coefficient for high conf calls are very high at 0.982. For the mutation data points that laid at the bottom of the graph, almost all of them are low conf or unclassified calls. Where they are on the x-axis is their VAF estimate based on the reference whole genome sequencing read counts. Where they are on the y-axis is the VAF estimate based on the validation platform's read count. So if they're on the bottom of the graph, it indicates that those variant calls were not validated in the validation platform, and there was no variant read in the validation platform. The top right figure is the whole exome sequencing on ion torrent at about 34x in depth. The diagonal oval shape is fatter because of low depth on the validation platform. The Pearson's R is expectedly lower at 0.93 because of it. There are a number of high conf calls at the bottom. Those are due to very low depth in the validation platform, such that their variant read was not discovered in the ion torrent data. The figure on the lower left is also whole exome sequencing, but on HiSeq. This may not really count as orthogonal because they are the same platform that produce the whole genome sequencing data, and the Pearson's R is close to 1. Nevertheless, almost all of the variants not validated are low conf and non-classified calls, and almost all of the high confidence calls are right in the middle where you expect them to be. The figure on the right, on the lower right, is PacBio long read data. The depth is pretty modest at 40x, with Pearson's R for high conf calls at 0.94. Like the other platforms, almost all the data off the diagonal are low conf and unclassified calls, and almost all of the high conf calls are where you expect them to be. This table shows the validation rates for somatic mutations in the high confidence reference call set and low confidence calls that we have removed from high confidence regions. For SNV, out of over 1,400 calls that had at least one validation platform resequenced, there was only one call that was deemed not validated by our threshold. That variant call had very strong evidence and many variant reads in almost all of the whole genome sequencing datasets, but no such read was found in the implicit data. We believe it was more likely a false negative in the implicit, a false negative in the validation platform, rather than a false positive in the reference call set. When there was contradicting evidence between 21 sequencing replicates in whole genome sequencing versus one single implicit dataset, we had very little reason to overturn it based on that single implicit dataset. For indels, the two calls that's deemed not validated were removed from the reference call set manually because upon inspection we saw a few variant reads in some of the whole genome sequencing replicates. We erred on the side of caution by removing those calls from the high confidence regions. For the low conf and unclassified calls that were deemed validated, we want to note that it does not necessarily mean that those are true mutations we have missed in the reference call set. They were deemed validated in this table because the read counts in the validation platform put them above that threshold, but many of them still carry sequencing features indicating that they are likely false positives. Here are two examples of unclassified calls that were deemed validated in the validation platforms. The photo on the left shows 3 screenshots of genome viewer for the variant in question. From top to bottom, it shows the BWA alignment for the tumor, the novel alignment for the tumor, and the BWA alignment for the matched normal. The variant call on the left may indeed be a somatic change. The tumor alignment indicates a substitution change 1 base pair away from a 13 base pair deletion. This may be a complex variant, though maybe not really a SNV. There are a couple of examples like this. Our reference call set may not have a comprehensive list of complex variants, though it includes many bi- and trinucleotide variants. The variant call on the right is almost certainly a false positive, even though it was quote-unquote validated in a validation platform. The variant alignment only existed in the BWA alignment and not in the novel alignment. Even in the BWA alignment, based on the coverage profile, it's quite clear that a deletion took place where the variant position was aligned. In the validation platform, we used BWA and we counted a number of variant reads that were misaligned. But this is definitely a false positive. As an example, we use a benchmarking software called HAPPY to benchmark the somatic mutation calling of STROGA2 in one of the WGS datasets with the highest sequencing depth among the 21 replicates, the Novartis replica number 2. It had sequencing depth of over 80x. The STROGA2's accuracy was very good on this dataset, with a sensitivity of over 97% and a precision of nearly 87%. 97% sensitivity means that 3% of the calls in our reference call set were missed. Of the somatic mutations not called by STROGA2 in this dataset, 60% of them are medconf calls, even though medconf calls make up less than 5.5% of the whole reference call set. This means that using STROGA2 in this dataset, we can recall almost all of the high-conf calls in the reference call set, but medconf calls were considerably more challenging. We are showing 1 false negative and 1 false positive from the call set in this slide. The false negative is shown on the left with 2 genome viewer screenshots. The screenshot on top is the 350x data from Genentech. The screenshot on the bottom is the Novartis dataset. There were 9 variant reads in the 350x dataset, but only 1 variant read in the Novartis dataset. So it was not STROGA2's fault that the variant wasn't called. There simply wasn't enough signal to call it in a dataset that had only 80x coverage. A false positive is on the right side. The screenshot on top is the 350x tumor dataset. The screenshot on the bottom is the 350x normal dataset. In this pair of 350x dataset, there were about as many variant reads in the tumor as there was in the normal. In the Novartis dataset, however, no variant read was captured in the normal. Therefore, it was called a somatic mutation there. This is an example of why we needed so many sequencing replicas to create the reference call set with such high confidence. The manuscript describing the work was deposited in bioRxiv preprint server. It was recently accepted by Nature Biotechnology. All the data are publicly available. All the FASTQ files, those are the raw sequencing data, are available on SRA. BWA aligned BAM files, reference call sets, and the high confidence regions are on NCBI FTP website described here. A lot of the documentations can also be found on this Google site. This work is only possible due to over 200 members from 60 institutions across government, academia, and industry. Thank you.